So dear students, so continuing from the previous topic that we are just understanding, or we are just going through the events after the fertilization or post fertilization events. So in post fertilization event we have divided the complete processes in 4 steps. The first one is development of endosperm, second one embryogeny or embryonic development, the third step is to go with the seed development, and finally some specific mode. So that we'll discuss later on. So now we are just moving on to the next one, that is what the development of embryo or embryogeny. So after completing the first step, that is what the embryogeny, now we are just after completing the first step, that is endosperm development, now we are moving on to the next step, that is called as embryogeny or development of embryo. What is the meaning of embryogeny? Embryogeny is a kind of process where the fully matured zygote or the zygote is going to convert in form of a fully matured embryo, so that it could further proceed in the process of seed formation and finally in the fruit formation. So 4-step process is there after the fertilization, you know very well now. Endosperm development, development of embryo, then the development of seed, and finally the seed will get converted in form of the fruits. So first of all, endosperm is now completely developed. Now the zygote will undergo in the process of converting itself in form of the embryo, so that it could further proceed in form of the seed development. So let us understand how the embryogeny occurs, or how the development of embryo occurs. So first of all, we are just going to understand the development of embryo in the dicot plants. So what is how the dicot plants are undergoing in the process of embryonic development, how the zygote is going to convert in form of a developed embryo. Just take care, just listen carefully. First of all, the embryonic development in the dicot plant was studied by Hanstein. Very important concept that I am just going to deliver. The dicot embryo, the development of dicot embryo was studied by Hanstein, in where it has been studied in Capsula bursa pastoris. So a study of dicot embryonic development was made by Hanstein in the plant Capsula bursa pastoris. Commonly this is called as the safford purse, and the Capsula bursa pastoris belongs to the cruciferae family. Are you getting my point? Who has studied it first? The Hanstein has studied it first in Capsula bursa pastoris, that is safford purse, and the plant belong to the cruciferae family. And if you remember, continuously we are taking the example of Capsula. Everywhere we have taken the example of Capsula, because this is the classical example, classical case where the embryonic development has been studied. Due to the process of embryonic development has been studied in Capsula bursa pastoris, and that belongs to the cruciferae family and Anagraceae. So that is why this type of embryonic development is also called as onagrade type or the crucifer type. What is it? Onagrade type or crucifer type. So here we are just going to start the process. You knows very well, in the embryonic in the development of embryo sac, we have made it clear. During the fertilization, the egg apparatus is going to convert in form of the zygote. The egg cell is going to convert in form of zygote, and the egg cell is lying toward the micropylar end only. So this is what the embryonic development will begin, where it will begin there in the micropylar end. So is it clear? Because egg cell and the zygote, the egg cell is fertilized in the micropylar end and it is being converted in form of the zygote there. So the embryonic development will begin there in the micropylar end. And one more thing, very important thing, we knows very well, the second male gamete fused to the polar nuclei and formed the endosperm. So the embryonic development will begin only after completion of the endosperm development, and that is what in the post fertilization changes we have studied first the endosperm development. The reason behind that, why the embryonic development will succeeded after the, or will succeeded by the development of endosperm, because endosperm is the source of nutrition. It is going to provide the nutrition to the plants. So is it clear how or why the endosperm development is preceded by the embryonic development? Because endosperm is going to provide the nutrition or energy for the process of embryonic development. So let us see how the embryonic development occurs in case of dicot embryo. I'm just going to make it clear with the help of flowchart, and later on we'll observe the process with the help of diagram also. What happens? The zygote lying on the micropylar end, we knows very well. When endosperm develops, it will start taking or driving the nutrition from the endosperm by increasing the nutrient content inside, by absorbing the nutrient content inside. You knows very well the size of zygote will get enlarged. The enlarged zygote now will get surrounded with the cell wall made of cellulose. What will happen? The enlarged zygote will get surrounded with the cell wall made of the cellulose. When it is getting surrounded with the cell wall, now the zygote is well prepared for the process of embryonic development and will start its division process. The complete embryonic development is the process of division and differentiation. Just take care of that. The complete process of embryonic development is just the process of division and differentiation. So what is going to happen after that? So when the zygote is going to be surrounded with the cellulosic cell wall, transverse division will be there. The division will be what? Mitosis. So everywhere, whenever or wherever the embryonic development is there, so which kind of division will be there? That is the mitotic division. So due to the mitotic division, the transverse division is there, and the transverse division result in the formation of two unequal cells. Just take care. Two unequal cell. One cell is called as basal cell, another one is called as apical cell or terminal cell. One cell is called as basal cell, another one is called as apical cell or the terminal cell. Basal cell is also known as the suspensor cell, while the apical or terminal cell is known as the embryonal cell. So from the name only it is very much clear. The basal cell is going to form the suspensor and the apical cell or terminal cell is going to form the embryo. So this is very much clear to you all. The two unequal cell is being produced. The basal cell is the larger one and the smaller one is the apical or terminal cell. So apical cell will continue in form of embryonic development, so that is why this is also referred as the embryonal cell, and the basal cell will continue in the development of the suspensor, so that's why this is called as what? Suspensor cell. So here I am just going to continue with the suspensor cell or basal cell first, then we'll move back to the, or we'll come back onto the embryonal cell. So just take we are just going to see the process what will happen with the basal cell. So just see, the basal cell or the suspensor cell are the larger cells which are lying on the micropylar end. So two cells are being produced. One is larger, one is smaller. So larger cell is lying toward the micropylar end, and that cell is called as what? The basal cell or the suspensor cell. So basal cell or suspensor cell are the larger in size, micropylar end they are lying, and now they are undergoing in the process of repeated division. What kind of division they will have? They will have the mitotic division, since the complete embryonic development is involved the mitotic division only. So after the complete mitotic division, they are just going to make a 6 to 10 celled long structure. What the basal cell is going to make the 6 to 10 celled long structure. The structure is called as suspensor. What is that? The 6 to 10 celled long structure is being produced by the division or repeated division into the basal cell, and that long structure is called as suspensor. So this is what the basal cell is called as suspensor cell. Why the basal cell is called as suspensor cell? Because this is going to form the 6 to 10 celled long structure, that is called as suspensor. Now what happened? The last cell which is lying at the micropylar end, the last cell, the terminal cell of the suspensor cell, the terminal cell or the last cell of the suspensor cell which is lying on the micropylar end is getting swelled up and enlarged in size and form a specific structure called as hastoria. Are you getting my point? The basal cell which is present near the micropylar end, the first basal cell which is present near the micropylar end gets swelled up, enlarged and form a specific structure called as hastoria. So now we are just going to understand the importance of these hastoria and the suspensor. These two structures are being formed. We'll see the diagram, then you will have the more clear ideas about that. The hastoria is the swollen structure which is known to absorb the nutrients, absorb the stored material. What is the function of hastoria? Hastoria is the swollen or swollen cell or enlarged cell which is known to absorb the nutrient, while the suspensor cell has a very important duty. By increasing its size, it is going to push the embryo there in the endosperm. What is the function of the suspensor cell? It is pushing the embryo into the endosperm. Why? Because endosperm is the source of energy for the embryonic development. So is it clear? The suspensor cell is meant for pushing the embryo there on endosperm cell, so that it could provide the enough amount of energy for the development of the embryo, and why hastoria is working to absorb the nutrient. One more important thing is there. the last cell of the suspensor lying toward the embryonal cell, the closer to the embryonal cell, the last cell toward the embryonal cell region or toward the chalazial end or toward the just closer to the embryonal cell is called as hypophysis. Are you getting my point? Last cell toward the embryonal cell is going to form a structure that is called as hypophysis. Hypophysis is very important because it is going to form the radical tip. Hypophysis, the importance of hypophysis or the function of hypophysis is it is going to make the radical tip. The radical tip is important to give rise the structure like root epidermis, root cortex and root cap. These all are very important point. Every time you have to get ready for the NEET examination. Here every time the questions may be there which structure is going to perform what kind of the function. So here hypophysis is known to give rise the root epidermis, root cortex and root cap. The suspensor pushes the embryo into the endosperm for the purpose of getting nutrition for the embryonic development, and haustoria is known to absorb the nutrients. So now everything is clear about that. So once again I'm just going to explain about the suspensor cell or basal cell. So suspensor cell or the basal cells are the larger in size. They are lying toward the micropylar and will undergo in the process of repeated division form 6 to 10 cell long structure that is called as suspensor, and that suspensor is work to push the embryo there into the endosperm. And finally the last cell that is lying toward the micropylar end is going to enlarge in the size, swells up and forms the haustoria. Finally the last one which is lying toward the embryonal cell is going to form the hypophysis, and it is responsible for formation of root epidermis, root cortex and root cap. Now after completion of the process of formation of suspensor and haustorium, now we are just going to return back toward the embryonal cell. If we have remember, the two unequal cells were produced. One is the larger that is lying toward the micropylar and is responsible for the formation of suspensor, haustoria and everything we have cleared. Now we are just going to see what happened to the next cell which is being formed, that is a smaller in size and lying closer or lying toward the chalazial end. And this is very much important question. If the question is there why the smaller cell or the embryonal cell is lying toward the chalazial end, so you know very well the chalaza is the place where parenchymatous cells are present, and parenchymatous cells are having the reserve source of energy. So that is very much easier to get the energy from there. So always the embryonal cell is lying toward the chalazial end. What happens now? The embryonal cell will undergo in the process of division. First division will be vertical or the longitudinal division, that will be followed by a transverse division. What will happen? The smaller cell or the embryonal cell or the terminal cell or the apical cell will undergo in the process of division. The division will be definitely the mitosis. No other division will be observed here. So first division in the embryonal cell or the terminal cell or the apical cell is the vertical or longitudinal division, that will produce the two-celled structure, and this two-celled structure will undergo in the process of another division that will be perpendicular to the previous one, means this will be a transverse division. So first division is vertical or longitudinal and second division is the transverse division. Due to that, now you can understand well, the first division result in the two cells and second division will form the four cell. So now the four celled embryonic stage is formed, that is called as the quadrant stage. Are you getting my point? By one vertical and one transverse division, now four celled embryonic stage is formed, that is called as quadrant stage. Now this quadrant stage undergoes in once again in the process of division. Now the division plane is the transverse. One longitudinal, one transverse form the quadrant stage, four celled embryonic stage. Now that quadrant stage will undergo in the process of transverse division once again, and if the transverse division again there, so the octant stage will be produced. 4 to 8 cells will be produced. So 4 celled stage is now going to convert in form of the 8 celled stage. At that 8 celled stage of embryo is called as octant stage. Now this octant stage, the 8 cells of this octant embryo or this embryo is getting arranged in two different tiers. Tiers means the group of the cells, two different tiers. They are arranged in the upper tier and the lower tier. The octant stage, the 4 cells arranged in the upper tier and remaining 4 cells arranged there in the lower tier. The upper tier cell is going to form the epiblast. The upper tier cell is going to form the epiblast, and the lower tier cell is going to form the hypoblast. The epiblast is responsible for the formation of plumule and cotyledon. The epiblast cell is responsible for the formation of plumule and cotyledon, and the lower tier cell is responsible for formation of hypocotyle and radicle. The lower tier cells are responsible for formation of hypocotyle and radicle. Radicle is going to give rise the root system. Plumule is going to give rise the shoot system. So what is there? First of all, one vertical, one transverse division, quadrant stage. Now one transverse division, octant stage. Octant stage, the cells will arrange into the two-tier structure. The upper tier is going to form the epiblast, which will give rise the plumule and cotyledon, and the lower tier is going to give rise the hypoblast, which is going to give rise the hypocotyle and radicle. So now what happens to the next? Now this octant embryo or octant stage embryo will start dividing periclinally, and after one periclinal division it will form what? A 16-celled embryonic stage. What will happen? One more division will occur there in the octant stage, and due to that one additional division the octant stage will divide due to one more division the octant stage will get converted in form of 16-celled embryo stage. Now the 16-celled embryonic stage is considered as the pro-embryonic stage or pro-embryo. Somewhere the octant stage is also considered as the pro-embryo, but this is not the mature embryo. Pro-embryo is primitive embryo that is going to convert in form of the advanced embryo or fully matured embryo. So 16-celled stage is going to convert is called as the pro-embryo. Initially the pro-embryo stage or the 16-celled stage is the globular in shape. Now the process of division and differentiation began. In the globular shaped embryo, the continuous repeated series of division will undergo, and along with the division the differentiation process will undergo. What is differentiation? Differentiation means what? The particular type of the cell will start behaving as a permanent cell. So during the differentiation what happens? First of all, the development of cotyledon and plumule is observed. During the division and differentiation what will happen? The growth of cotyledon with the plumule. Cotyledon and plumule growth will be there, because cotyledon will be used as the source of energy for the embryonic development, and plumule is going to give rise the shoot system. And as soon as this differentiation is going to start, the cells are started moving toward the particular structure, toward the particular area, and a heart shaped embryo is formed. Remember, in the NEET examination, first embryonic shape is globular shape or globe shaped structure, and later on it is going to convert in form of a heart shaped embryo. So now very next to that, when they are just going to convert in form of heart shaped embryo, the differentiation process again began. During the differentiation process what happened? They are just going to prepare a kind of permanent tissue generating structure or permanent structure which is going to produce the particular tissues. The structure those are going to develop are called as histogens. Histogens are the specific tissues those who are having responsibility to develop or generate the particular type of the tissue. Histogens are the particular structures or tissues which are going to generate the particular type of tissue. Here three histogens are being developed. The first one is called as dermatogen, second one is called as periflame, and third one is called as plerome. Three basic histogens is being produced there in the heart-shaped embryo after the process of differentiation: dermatogen, periflame, and plerome. Dermatogen is known to produce the epidermis. The dermatogen will form the epidermis in the later stages. Now periflame will form the cortex, and the plerome is responsible for the formation of stele. Stele is a specific structure which is formed of the vascular tissue, ground tissue, and the pericycle. It is a kind of vascular tissue which is present in the centermost region of the plant tissue. So this is going to form what? Stele is made of what? Vascular tissue, that is xylem, phloem, and cambium. The ground tissue is made of pith, and the pericycle. So this is what the complete embryonic development is now being completed till now. One more important thing, the embryonal axis concept is there. Embryonal axis is the axis from where the embryo is being developed. That embryonal axis is having two different regions. Embryonal axis is divided into two parts. One part is called as epicotyle, another one is called as hypocotyle. So what is epicotyle? The part of cotyledon, the epicotyle is the part of embryonal axis which is above the cotyledon. What is epicotyle? Epi means upper, cotyle means cotyledon. So epicotyle means the part of embryonal axis which is present above the cotyledon, and this is going to terminate in the plumule. Are you getting my point? Epicotyle is the part of embryonal axis which is going to terminate in the plumule and present above the cotyledon, and hypocotyle is the vice versa. Vice versa means to say this is the part below the cotyledon. Hypo means below, cotyle means cotyledon. It is the part below the, it is the part of embryonal axis below the cotyledon, and which terminates in radicle, not in the plumule. So now this is clear to you all what is happening there in the embryonic development. The embryonal cell is going to divide first time by the vertical division, then transfer division. Quadrant stage is there. Now the transfer division will form the octant stage. The octant stage is once again going to divide, octant stage is once again going to divide transversely and form the two-tier arrangement of the cell, epiblast, hypoblast. Now epiblast, hypoblast once again divide into the once again they are going to divide and form the 16-celled embryo. Now the embryo is pro-embryo, then division, differentiation, heart-shaped embryo is formed, and 3 different histogens are being formed: dermatogen, peribleme and plerome. So now we are just going to understand the entire process by the mean of, or by the use of diagram. So just take a look on. Here is the diagram of the embryonic development of a typical dicot embryo, that is of the capsular type. What happens here, you can see the zygote is there, diploid structure, will start absorbing the nutrition, will get surrounded by the cellulosic cell wall, will undergo in the process of mitosis. Transverse mitosis will be there. One cell which is lying towards collagen will be the smaller in size, and which is lying toward the micropyle will be larger in size. So the cell which is lying toward the micropylar end is known as CB cell, basal cell, CA cell apical toward the collagen end. So now what happens, the division will occur there in the basal cell, that I have just cleared you. The repeated series of division in the basal cell will get form a kind of 6 to 10 cell long structure that is called as suspensor, and the cell lying toward the micropylar end will get enlarged, swells up so that it could get absorb the nutrition from there. So it is clear to you all, haustorium is known for the absorption of nutrient, and the suspensor cell is having function to push the embryo there into the endosperm for getting the nutrition there. And one more important thing was there, hypophysis. We knows very well, the cell which is lying closer to the embryonal cell and is having responsibility to develop the root structures. So now moving on to the next, what will happen to the apical cell or the terminal cell? We knows very well, first vertical division here, then the first vertical division, then the transverse division, first longitudinal division, then the transfer division. Now the quadrant stage is formed, that is 4-celled stage. Afterward what happened, one more division occurs, and that is going to convert in form of what, an octant stage or 8-celled stage of the embryo. An 8-celled stage is called as octant stage. The upper tier of the cell is going to form the epiblast, and lower tier of the cell is going to form the hypoblast. So very next to that, what happened, the continuous division, and going to a continuous division is going to form the differentiation and the change in the shape of embryo. The embryo is getting converted in form of the heart-shaped structure. The heart-shaped structure, you knows very well what happened. First of all, the development of cotyledon occurs along with the development of plumule. So here plumule is there, and this is what the cotyledon, the plumule is there, and this is what the cotyledon. Along with that, the suspensor cell lying at the top, root cap is present here, and 3 basic histogens are being produced, and the peripheral region, the dermatogen is present. Just below the dermatogen, peribleme is there. Just below the peribleme, the plerome is there. So 3 different dermatogen are being produced. So what happens in the pro embryo, now it is going differentiated in form of the heart-shaped structure, and due to continuous differentiation, what will happen, the plumule is going to form and the cotyledons are going to form. Along with that, 3 histogens are being formed, that is what, dermatogen, peribleme and plerome. Finally, the embryo is getting converted in this shape of the structure, where in the top only the haustorium, you knows very well, the suspensor, the root cap, this portion defines the radicle. Now this is what the embryonal axis. This portion is called as hypocotyle, this portion is plumule, and this is called as what, cotyledon. And this is what, the embryonal axis. So this is hypocotyle and this is epicotyle. So remember my dear child, this diagram is asked repeatedly. This one, a dicot embryo diagram. This is the diagram of a mature dicot embryo. This is the diagram of a mature dicot embryo, and many of the time this has been asked not to draw the diagram but to label the diagram. Many of the structures are there and these structures are being asked with their function. The questions may be there like that, okay, how just list down or label the structure 1 2 3 4 and provide one function of each of the structure, label the structure 1 and 3 and provide the function of each of the structure. So like that, you know very well. Haustorium absorbing the nutrition, suspensor pushing the embryo in the endosperm region so that get the nutrition, root cap protection of the root, radicle root system development, embryonal axis upper side is epicotyle, lower side is hypocotyle, plumule is going to give rise the shoot system, and these are the cotyledons. They are providing the additional material for the embryonic development. One very important thing that is I'm going to discuss here, that is what the development of embryo in capsula. When it has been observed in the capsula, the embryo is arranged in form of the curved position inside the ovule. The embryo of the capsula arranged in form of the curved position inside the ovule. This position of embryo is called as torpedo. Remember, in capsula only the embryo is like that. The embryo is arranged in the curved position and this condition is called as torpedo. So everything is clear to you all now. Zygote divides mitotically. Larger cell toward the micropylar end will form the basal cell, smaller cell toward the chalazial end is form the apical cell. Division into the basal cell forms suspensor and haustorium. Now the embryonal cell, vertical division, transverse division, 4-celled stage. Now the epiblast, hypoblast, octant stage, continuous division, maturation, proembryo, heart-shaped stage. Then the development of plumule, radicle, everything, and this is what the structure that you have to take care. You have to go with that. Every structure with a function, you need to take care of that. So this is what about, or this is all about the development of a dicot embryo. Now after completing the development of dicot embryo, now we are moving toward the development of a monocot embryo. How the monocot embryo is being developed. So there is not much difference in the pattern of development of monocot or dicot embryo. Only the matter is of the monocot and dicot. There two cotyledon is developed and here single cotyledon is developed. The basic process is quite simple and the simpler mode of development is also. There is not a difficult mode of development. A very simple mode of development is there. So what will happen? As usual, zygote enlarges by getting the nutrients. When it enlarges, will start its division. The transverse division will be there, and due to the transverse division, as similar to the dicot embryo, the two type of cell will be formed. One is the larger and one is the smaller. The larger cell is called as CB cell, basal cell, CB cell, basal cell, larger cell lying toward the micropylar end, and the smaller cell is called as terminal or cell apical cell. So is it clear? The terminal cell and the basal cell, apical cell and the basal cell. Basal cell is the larger cell and the terminal cell is the smaller cell. Okay. So what happens? In the basal cell will start their transverse division. Basal cell will start their transverse division and will form the two cells called CI and M. What will happen? The basal cell will undergo in the transverse division and will form two cells called as CI and M. At the same time, the embryonal cell will once again start their division. The embryonal cell will undergo in the process of longitudinal division and will form two-celled stage. The two-celled stage will undergo in the process of transverse division and will form the quadrant stage. Here there is no need of forming the octant stage because the embryo is monocot embryo. So what is happening? The first of all, the basal cell is going to divide transversely and form two cells CI and M, and then the embryonal cell or the terminal cell is going to divide first the longitudinally and second time transversely and form a quadrant stage. The cells are being arranged into the two tiers, the two cells arranged on the upper tier and the lower tier. These tiers are here notified as L and L dash. The quadrant stage cells are being arranged into the two tiers called as L and L dash. So at this stage, the embryo is considered as the proembryo stage. The 4-celled embryo is considered as the proembryo, and now the embryo is looks like an inverted T-shaped embryo. What is the shape of embryo? The embryo is looks like an inverted T-shaped embryo. The embryo is an inverted T-shaped embryo. Now what happens, the cells, what are the fate of the cells here present, just we are just going to see. We have seen the after longitudinal division, the quadrant stage of the embryo is having two type of the cell, L and L dash. The L cells are going to form the portion of embryo below the cotyledon. The portion of embryo below the cotyledon is formed by the L cells or cells of the L layer, and the L dash is going to form the cells above the cotyledon. Means what? L dash is the upper layer and L is the lower layer. So L dash is going to form the cotyledon, hypocotyle and plumule, and similarly the L is going to form the epicotyle and radicle. So what is there? L dash is the upper layer of the cell which is going to form the cotyledon, plumule and hypocotyle, while L is the lower cell below the cotyledon is going to form the radicle. So that is going to form the root system, and L dash is going to form the shoot system. Now we are moving toward those two cells which are remaining there, the Ci and M cell, that is being produced by the transverse division of the basal cell. So Ci once again undergoing the process of transverse division and will form two cells. The cells are called as N and N dash. The Ci undergoes in the process of transverse division and will form two cells, N dash and N. N dash is the upper layer of the cell and N is the lower layer of cell. The N dash is going to form the suspensor while the N is going to form the root cap. Very simple process, not difficult like the dicot embryo. So Ci is going to divide transversely to form two cells, N dash and N. Upper layer is N dash, form the suspensor, lower layer is N, form the root cap. Now the second one, M, what the M does? So M is going to form the root cap and the perivlem. So is it clear to you all? What is zygote divides transversely unequally, basal cell and the terminal cell. Basal cell is larger one, terminal cell is the smaller one. Larger cell will divide transversely to form the two cells Ci and M, and terminal cell is going to form the two cells, first of all longitudinal division, then the transfer division, two cells L and L dash is there. And now at that stage the embryo looks like the inverted T-shaped stage. The portion below the cotyledon of the embryo, the portion of embryo below the cotyledon is expressed by the L, and L dash is above the cotyledon, so makes the plumule and the shoot system, and L is going to make the root system and the radicle. Ci divides one again to form the N dash and N. So N dash is going to form the suspensor, N is going to form the root cap, M is going to form the root cap and perivlem. Okay, if we could observe the diagrams of the embryonic development in the monocot embryo, here the simply the zygote enlarges in the size, the transverse division is there, two cells Cb and Ca. Ca is the apical cell toward the chelagel and the smaller, Cb the larger one. Now what happens? Cb is going to divide transversely into the two cells Ci and M. These two are the cells of the Cb. Actually I just made the diagram with the help of color so that you can understand. The green color is for the Cb and red color is for the Ca cell. So now what happened? This Ci cell is undergone in the process of division and form N dash and N. These two are the cells produced by the division of Ci cell. M remains as it is. So now this complete structure is produced from the Cb. So N dash you know very well, suspensor, haustorium, everything we have seen. So N dash is going to form the suspensor and root cap. Then M. Now embryonic cell, the single cell is there. First vertical division or longitudinal division, then longitudinal and transfer division, quadrant stage, they are just going to form the L dash. L we knows very well what is there. Now this N dash forms the suspensor, then root cap, and this portion is going to form the radicle. After the radicle, hypocotyle is the next one, then plumule and the cotyledon. Cotyledon is the single, there is no differentiation, so that is why the heart-shaped embryo cannot be formed. Here the globular embryo is there. Finally the embryo or mature monocot embryo is seems like that or looks like that. The suspensor, the hypocotyl region, this region is of the plumule or stem region, the cotyledon. So this is what the complete mature embryo of the monocot. So I think now everything is clear to you all. Should I go for repeating the complete concept of the today? So one by one I'm just going to repeat the complete concept of today. So in the dicot embryo, crucifer type, onagrade type, cephod purse में study किया था, that is in capsula bursa pastoris by Hanstein. The process zygote divides after getting nutrition. Two unequal cell, one is basal cell toward the micropylar end, the larger cell, another one is the smaller cell, that is apical or terminal cell toward the embryon, toward the chalazial end, and called as the embryonal cell. Embryonal cell responsible for developing the embryo, and the suspensor cell is responsible for developing the suspensor. Suspensor cell will undergo in the process of repeated division and form a 6 to 10 cell long structure called as suspensor. The suspensor is going to form what? Suspensor is going to push the embryo into the endosperm. The last cell near the micropylar region is going to swell up and form the hastoria to absorb the nutrition. The last cell toward the embryonal cell is called as hypophysis, which is going to develop the root tip, and it is responsible for developing the root epidermis, root cortex, and the root cap. The smaller cell is embryonal cell. Vertical division 1 transverse division, quadrant stage, once again transverse division, octant stage. Octant stage, the cells arranged into two tiers, epiblast, upper tier, lower tier, hypoblast. Epiblast is going to form the plumule and cotyledon, and the lower tier is going to form the hypocotyl and radicle. Means epiblast is responsible for development of shoot system, and the remaining part, that is hypoblast, is going to produce the root system. Then one more division, 16 celled embryo, globular shaped embryo, division, differentiation, development of the cotyledon and the plumule. Finally the heart shaped embryo, development of the histogens, that is dermatogen for epidermis, periblaim for cortex, plerome for the stele. Embryonal axis, we have seen the embryonal axis. The two part of embryonal axis is there: epicotyle, the part above the cotyledon, epicotyle, which terminates in form of the plumule, and hypocotyle, the parts below the cotyledon, terminates in form of the radicle. Diagrammatically, zygote, apical cell, basal cell, suspensor cell, embryo, 2-celled stage, 4-celled stage, octant, so epiblast, hypoblast. Now division, differentiation, the cotyledon, plumule, dermatogen, periblaim, plerome, suspensor cell, root cap, and this is the final structure. This is very important, and always this is asked to relabel the diagram, and even the function is being asked. Hastoриum, you know as well, driving the nutrition, suspensor pushing the embryo into the endosperm, root cap protecting the root, embryonal axis, development of embryo, radicle, root system development, hypocotyle, epicotyle, plumule, shoot system development, cotyledon, reserve food energy. In monocot embryo, the zygote, enlargement, transverse division, CB cell, cell basal, large transverse division, 2 cells, CI and M. In the terminal cell are the embryonal cell, the small cell, CA, apical cell, longitudinal division, 2 cells, and transverse division, 4-celled, quadrant stage is being formed, L and L dash, 2 arrangement is there. The shape of embryo is inverted T. So 2 cells, L is going to form the layers below the cotyledon, and L dash is formed the layer above the cotyledon. So that will going to form the plumule, while L is going to produce the radicle. And here CI is divided once again in form of N dash and N. N dash is going to form the suspensor, and N is going to form the root cap. M is going to form some part of the root cap and the periblaim. In diagram, zygote, enlarged, transverse division, CB, CA, CB divide CI and CM, then CI divide N dash N, N dash suspensor, root cap, M is going to form the periblaim as well as the some part of the root cap. So this is the complete differentiation of the CB cell. Now the CA divide longitudinally, then vertically, the quadrant stage, L dash L, L dash hypocotyle, plumule L, radicle, and the epicotyle. Radicle is going to be produced thereon. So here what happens? Suspensor, root cell, First of all, radicle, then hypocotyle, the plumule, and cotyledon. Single cotyledon is there, that is why no heart-shaped structure is formed. So this is the matured embryo of the monocot, suspensor, hypocotyle, stem embryo. So this is what. So now we have completed the development of monocot and dicot embryo. So now major portion is now completed. Now tomorrow in the next lecture we'll continue with the development of seeds, ki how this embryo is going to convert in form of the seed. Now very easy portion is there. So tomorrow, till the time, thank you.